One right here, the newest addition to this list, unfortunately, is this species right here, Palmer amaranth. We now have glyphosate-resistant Palmer amaranth in Illinois, and for those of you who don't know this species, this is the one that's changed farming practices in areas of the Mid-South and the Southeast. If these populations become more widespread and rampant in Illinois, folks, we have a serious problem. The one that we contend with and talk about the most is this one right here. This is Waterhemp. And this is a species that 20 years ago, barely 2 people in the state of Illinois even knew what it was. And now it's hard and difficult to find 2 people involved in production agriculture in the state who don't know what this is. Now the challenge, of course, with Waterhemp is the number of different types of resistances that it has evolved. Right now in Illinois, unfortunately, we lead the United States in the number of different types of resistance that we have in the Waterhemp populations around the state. We actually have 5 different types of resistance in Waterhemp populations in Illinois. So if you as a farmer, for example, would have resistance to any one of these, it's not just whichever one is more important, of course, but resistance to any one of those things would introduce challenges into your production system. But unfortunately, that's not where our story ends with respect to Waterhemp, because this is basically where we are right now in various parts of our state. And that is, how do you effectively manage this species or this population of this species when we do not have any effective herbicide options for its control once it comes out of the ground? And the rationale behind this is this phenomenon right here. Remember we said we had now resistance to 5 different chemical families in Waterhemp populations around the state. But unfortunately, that's not the most problematic phenomena occurring with Waterhemp. It's this. And this is what happens when this species begins to stack those types of resistances within individual plants. This is one that we actually identified from over at Adams County, Illinois. This is just a comparison of a sensitive population that's actually sensitive to these 3 different chemical families. This plant right over here is resistant to all 3 of them. So it's not a matter that it's just resistant to a triazine herbicide or a protox inhibiting herbicide or an ALS inhibiting herbicide. All 3 of those are ineffective against this particular plant or this particular population. This is where we're headed to. This is where we're going. And the reason that we maintain this is actually based on survey data that we've actually done over the last several years. And these are from 2010 and 2011 surveys that people would send in to us for resistance screening. Now, over these 2 years, we've had 512 plants submitted from 121 different fields, mostly in Illinois. There have been a few that have snuck across the border, but we just tell folks in Iowa that we do really like them anyway. But we're looking at the different types of resistances here, 3 different classes of herbicides, the ALS inhibitors, the PPO inhibitors, and the glyphosate. And what we see on here is that overall, out of these 512 plants, only about 12% were sensitive to these herbicides. Now in these Venn diagrams, you can see the percentage of resistances, say, to glyphosate. About 61% of those plants were resistant to glyphosate, 16 to PPO, 56 to ALS. Now any of those, again, can be problematic from a very practical standpoint, but when you look in the overlapping areas of those Venn diagrams, you begin to see the story unfolding. So about 25% demonstrate resistance both to glyphosate and to ALS, 2% to PPO and glyphosate, 5% are 3-way resistant. We actually, I didn't mention it yet, but we actually have at least one population in the state, I think we just submitted the manuscript last week, that's not 3-way resistant, it's actually 4-way resistant. We'll get to a point where we see 5-way resistance and perhaps even 6 before it's all said and done. On a field basis, of the 121 fields sampled from, less than 10% were free of resistance. Okay? So 91% of the fields that were sampled actually did contain at least one type of resistance in that field. In our group, when we deal with resistance, we have, I guess, what I would like to consider one of the most effective working groups that we have virtually at any land grant across the Midwest, because of the different areas of expertise that we have in order to address these problems. And I'll show those names here in just a moment. But with respect to resistance, a lot of what we do includes field, greenhouse, and laboratory research. And we have faculty members now who have expertise areas in the molecular biologies that really have opened up new avenues that we can look at that when I arrived here about 20 years ago, we could only dream about. So we can do things like the identification and screening for resistance. We've actually evolved this particular facet over the years. It used to be screening involved obtaining seeds from suspected populations, growing them up in the greenhouse, overspraying them, and more or less just evaluating the results and getting those back to whoever sent the sample to us. It's a very time-intensive process. It may take 6 months to get an answer back. But now with some of our colleagues in molecular sciences, we can basically now use molecular biology techniques and have these answers within 24 hours. With a very, very improvement on the system. Management of resistant populations, of course, based a lot on what we do in the field, but as well incorporating the results from the laboratory and the greenhouse work, investigating and discovering resistance mechanisms. An area that we really had not really been able to do much with, again, before the arrival of some of the colleagues and the faculty that we now do have in the program. We also are now beginning to spend more and more time looking at new herbicide-resistant crop technology, one from Dow AgroSciences that will be introduced, another from Monsanto within a few years, trying to say how well these technologies will fit into the system. How can we utilize these new technologies to try to manage some of these resistance problems and yet at the same time preserve the effectiveness of that technology? Because we're dealing with species, obviously, that have demonstrated almost no problem with evolving resistance to various types of chemistries. And then simply looking at the invasive species that we have that we are concerned with, things like Palmer amaranth, for example, but also improving weed management in various cropping systems, perhaps with different types of options for herbicide delivery. These are the faculty that we have in our department in crop sciences at the University of Illinois. Dr. Tranel couldn't be here today. He's a molecular weed scientist, been on faculty now since I think about 1997. Dr. Dean Riekers works in weed and herbicide physiology, also with some very good molecular biology skills. Dr. John Masunis does quite a bit of practical and applied work in vegetable and organic production systems. Myself, primarily field and greenhouse research as well as extension and outreach activities. And I would be remiss if I did not mention our colleagues with the USDA ARS, who we also collaborate with on multiple projects. Dr. Adam Davis, weed ecology, landscape level modeling. He's been intimately involved with us on several projects dealing with resistance to help us better understand some of the factors involved in selecting for these biotypes, not just on the field scale, but typically even on the landscape level scale. Dr. Marty Williams works a lot with vegetable crops, sweet corn, weed management, and then Dr. Joanne Shee Sanford, microbial ecology of weed seed banks. So with that, that's basically all I had. So if there's, Greg, is there time for questions, I guess, now?
Yeah.
Or no?
Yeah, we have about a minute or 2 minutes.
Okay.
I think one of the things we were trying to do is we had a meeting with Romark last week and trying to make it connected with where the faculty in nanotechnology here are going to connect with to advance this in a way that we couldn't without that connection. Sure. So I think that the next point is to the faculty here to think about ways this might work together.
Yes, please.
Do you do a lot of sequencing work? I mean,
yes. Dr. Tranel does a lot of the sequencing work, either on a specific gene that we are interested in, or he's actually involved in a genomics project to try to sequence the entire genome of some of these amaranthus species.
Do you do that on campus or elsewhere?
Yes, on campus. Yes, sir.
Are there particular points in the research where there are limitations, either in terms of how quickly you can get assay results back or being able to do field deployable results?
Yes, the greatest limitations of the assay are simply having everything in place when we need to, because of the cost of that particular set of reagents that have to be used. We can't just run the assay when we get one sample in. So it may take a few weeks of acquiring enough samples before we have enough to justify running those particular assays. Yeah. That's probably the greatest bottleneck that we have. Yes, sir.
So my question is, what kind of weedicide applications do you use?
What kind of what?
What kind of application you use to control the weeds? And to what I see this as, because spray drift and all these are issues
Sure.
So where the nanoparticles or nano-encapsulated particles can be used on demand or specific smart spraying technique would be a potential where nanotechnology can complement with
To your first question, the unfortunate consequence of this multiple resistance in these either fields or individual plants is that we're running out of effective options. If they emerge out of the soil, we may, and right now we have those populations, we do not have something that you simply pour out of a jug to control it. With respect to the other, with the application technology, certainly with the forthcoming introduction of some of these new varieties, that is something that a lot of people are very, very interested in, because these products can be effective on these particular resistant populations, but keeping them in place is going to be very, very challenging given the fact we've got a lot of acres that have to be covered in the state in a relatively short period of time. Yeah.
Through the root system, through the leaves, for these herbicides, can you just enlighten us a little bit?
Sure. The mechanism of action would actually vary depending on the particular herbicide. Some are taken up primarily through the plant foliage after it emerges from the soil, whereas others are actually absorbed primarily through the root system of the very young root after the radicle actually ruptures from the seed itself. Okay. Thank you.
Thanks very much.
Microphone.